here for a video on skeletal muscle organization and cellular structure, just the basics of it. Skeletal muscle is one of the two types of striated muscle. Striated means striped. The other type being cardiac muscle. And the stripes actually originate, you can't see them except under a microscope. Histologically, the stripes show up because of the presence of sarcomeres. And as we'll see in a subsequent video, the sarcomeres, I might point them out here, but more on them in a later video. The sarcomeres are sort of the contractile units formed by proteins, the intracellular proteins of striated muscle. So what I want to do now is show you how skeletal muscle is organized and talk a little bit about the cellular structure. Skeletal muscle cells, often called skeletal muscle fibers because they're very long and skinny cells. I don't want to call them skinny because they're really big cells in general, but they are very long cells, so they're fiber-like. And they are formed by cell fusion. Now, this is not true of the cardiac muscle, only of skeletal muscle. And what that means is during development, the stem cells sort of fuse together and form bigger and bigger and bigger cells, these long fiber-like cells that have potentially hundreds of nuclei and thousands of mitochondria. So they're sort of monster cells. And that process of multiple cells fusing to form sort of these giant cells forms what's called a true syncytium. We had heard about functional syncytia before when we talked about cells that were connected by gap junctions. Their cells are working together as a unit, but they don't actually fuse. In this scenario, a true syncytium, the cells actually fuse and become one giant cell, essentially. Okay, so skeletal muscle is actually comprised of two tissue types, right? The skeletal muscle cells themselves and connective tissues. And so we have to see how the connective tissues sort of fit into the apparatus for generating tension. Skeletal muscles functionally, almost all of them are attached to bone, not all. There's a few in the face that are not directly attached to bone. But most of them serve to generate force to move our skeletons, right? To provide us with physical movement. It's one of the few cell types that's actually adapted to generate force, enough force to move other cells. And so I'm going to draw sort of a little tiny muscle up here at the top so we can sort of see what my next image is going to be. And so if we draw a muscle, right, and the blue ends here would be the tendons that would attach it to bone, right? And the meaty part here in the middle is called the muscle belly. If I were to cut through this muscle, right? And oh, by the way, the connective tissues, the tendons at the end are formed of dense regular connective tissue, if you recall. Really good for linear strength, for pulling, right? And so our muscle is going to be attached to our two endpoints, the bones that it's intended to move by those tendons. But the connective tissues of those tendons actually permeate an extension of them, if you will, or what's connected to them. There are more connective tissues that run through the muscle. So if we cut this muscle sort of right down the middle, right? And then we turn it, right? So you're looking at the cut end of that muscle. Let's do that. And so let's show that up here. All right, so if this is that cut end of the muscle, we can have the rest of the muscle sort of going off into the distance here behind. All right, and we had a blue tendon at the end of that. We'll add some blue tendon back there. So hopefully that makes sense. Right, so we've got our cut end of the muscle looking at the end of it. And the muscle is organized, many muscles are organized where it's not just you know, a whole bunch of cells, right? The muscle fibers themselves will be running in and out of, like, lengthwise through this muscle, right? So the muscle cells can be as long as the muscle that contains them. So they can be quite long. You've got muscles that are more than a foot long, some of the leg muscles. And so there are muscle cells, muscle fibers that are essentially the same length. Now, so those cells would be running sort of, you know, we would have cut through the cells and we're looking at the ends of the cells now too. But those cells are grouped in bundles called fascicles. And so I'm going to draw these bundles. That's a fascicle. So a fascicle is just a bundle of the individual muscle fibers. And so if I draw the muscle fibers now within those, right, right, and those would be the individual cells cut within each fascicle. And the fascicles would be jammed closer together than that. There's not this sort of empty space out here. But the connective tissues, as I mentioned previously, of the tendon sort of, you know, are continuous with connective tissues within the muscle. And so there is a connective tissue layer around the outside of the whole muscle called the perimysium. I'm sorry, not the perimysium, the epimysium, sorry. There is a connective tissue layer surrounded, so the epimysium wraps the whole muscle. And actually, there are connective tissue layers outside of that too that are called some of the fascial tissues. And there's a blurring of terms here, but we'll just go with the connective tissue wrapping right on the muscles, the epimysium. The connective tissue wrapping that separates the fascicles from one another is called the perimysium, right? And so around each fascicle is the perimysium. Right? Mysium refers to muscle. Myo means muscle. And then there's a connective tissue wrapping around each individual cell. Right? So those are the muscle fibers, the little red circles if you can make them out. Right? That's the cells cut in cross section. But each one of those cells is wrapped by a connective tissue layer called the endomysium. And it's important that I point out the importance of these layers. They're not just sort of separating the cells and the fascicles from each other, because those connective tissue wrappings are all continuous, end to end, along the entire muscle body. Well, the cells don't continue in, the muscle cells themselves don't continue into the tendons. But think about it. The tendon is sort of there attached at the end, and it is continuous with all of these wrappings, the endomysium, the epimysium, the perimysium, all the way at the end of the, sort of like the tendon is sort of like this bundle of all of those connective tissues sort of leaving the end of the muscle, right? So in that way, the connective tissues aren't really like just stuck to the ends. They permeate throughout the whole muscle. And that's really important because one of the key factors that has to happen is the muscle cells are contractile, but they're not attached to bones. Right? Their membranes aren't attached to bones. They are not directly pulling on bones. And so those muscle cells contract, and across the membranes of those cells, we have to have connections between the intracellular generation of force and these extracellular connective tissue protein layers of the endomysium, epimysium, perimysium, right? To sort of convey our tension all the way to the ends where the tendon will attach us to bone. Right? So those connective tissues are really important for the conveyance of force from the contracting muscle cells to the endpoints of the muscle. And they're so the muscle cells are going to interact with that connective tissue, especially directly with the endomysium, because that's the one right around it individually. But right, so we're going to have those connections between the cells and their endomysial layers that'll help convey force from cells to connective tissues, and then finally to bones. Okay. If we look in an individual cell, these giant individual cells, I'm going to point out some of the key cellular structures. And actually, before I do that, I think I could maybe do it here. One of the things I want to point out is this structure called the motor unit. And let me pick a color. I think I'll just have to go with blue. I don't have very many good colors here. So muscle cells are innervated by the somatic motor neurons and stimulated to contract for us to generate movement, right? So they're consciously generated motor movements involve motor neuron action potentials stimulating these muscle cells. And so we have to have synapses between the motor neuron and the muscle cells. And those motor neurons don't synapse with one muscle cell. They synapse with, as the minimum number is probably about a dozen, and upwards of possibly hundreds, maybe even more than 1,000 individual cells. And so what that means is we've got this motor neuron. I'm going to just I can draw a sort of mini brain and spinal cord over here, right? We're going to have a motor neuron that comes out of the spinal cord or the brain for controlling, structures in the head. But that motor neuron, somatic motor neuron, is going to leave, send an axon out, and it is going to come to the muscle where it's going to branch and branch and branch. And I'm going to just show this one with 3 branches. And I apologize if that's a little small. Let me put my muscle cells here, right? And so that combination of somatic motor neuron coming out of the spinal cord in this scenario and innervating individual muscle fibers is called a motor unit. So it's the motor neuron plus innervated muscle cells. Right? And so that is sort of the individual controlled unit within a given muscle. Right? We're going to have, and again, we don't have one motor unit per muscle. We have many of them, even though they contain, each one of them can contain dozens or even hundreds of individual muscle cells. Now, the key thing to think about here, though, is that when a motor neuron is stimulated and we reach its threshold, we're going to get a nerve impulse, right? With this repeating wave of action potentials being generated, right? And so we're going to get action potentials. We're going to travel down that axon. And again, there's another video that sort of shows how that stimulates the muscle fiber. But when it reaches the end of this this motor neuron, that motor neuron branches, which means a single action potential in one motor neuron will stimulate dozens, if not hundreds, of individual cells. So all of those cells are going to do the same thing at the same time, right? That is the key with a motor unit. Once you activate a motor unit or groups of motor units, each one of those motor units will be doing the same thing, right? If this one is generating an action potential every second and another group is generating an action potential every 3 seconds, those 2 groups of innervated muscle fibers are going to be synchronous with the 1 per second or the 3 per second or the 10 per second or whatever the rate is of their own motor neuron. Okay, so the motor neuron is sort of the functional unit of control for skeletal muscle. Okay, let's look at the individual cells. So let's go down here all the way to the end, and we'll blow that up a little bit so you can sort of see where that innervation occurs and what, again, not getting into the mechanics of the stimulation, but where that occurs and the other structures associated with the subcellular structures to generate tension, right? And so if we draw, drop my red. If we draw an individual muscle cell now, these are odd cells. Right, and the other end of the cells over here. it's got these sort of indentations of the membrane. And so our cell is going off, right, farther in that direction, farther in this direction. This is the big, long, cylindrical cell. And it's got the, we've sort of taken a cross-section of it here, so a longitudinal section, I guess I should say, where we're sort of looking inside the cell along its length. And we see these, indentations. They're actually called Ttubules, and the T is for transverse, meaning they are transverse to the direction the cell is around, the lengthwise direction of the cell. That's a Ttubule. I'll tell you what those are about in a minute. Now, we had our motor neuron innervating it, so we have to make sure we have a synapse there. Add that. Right, so here comes our motor neuron axon and the synaptic bulb. Right, it is going to contain, right, it's going to have synaptic vesicles down here. And the neurotransmitter released is acetylcholine. It's always acetylcholine at skeletal muscle. It's called the neuromuscular junction, right? Because it's a neuromuscle junction, right, between the somatic motor neuron and the skeletal muscle fiber, right? Okay, now, so we're going to have, again, more details about the mechanism later, but because of this, that means, because we're releasing acetylcholine, that means we're going to have ligand-gated channels, right? And so if I put channels here, right, they're going to allow ion flux, and we're going to generate, we'll talk about that later, an EPSP, right? That's stimulatory. It's a depolarization, so we know it's probably sodium or calcium, and it's sodium. But so that EPSP is stimulatory, and we can generate an action potential in this membrane because of the presence of voltage-gated channels. So I put a separate set of channels, right? And these would be everywhere. Put these sort of along the length. And again, both sides. Right? But those would be voltage-gated channels, voltage-gated sodium and potassium channels. So in other words, this membrane, this muscle cell membrane, is like an axon because it has voltage-gated channels, and because that, it can generate action potentials. And so this EPSP is going to activate those voltage-gated channels. We're going to get an action potential. Well, that's going to be our stimulus of the muscle cell. And that action potential, right, is actually going to travel along the membrane just as it did in the axon. And that's going to be our stimulus. Now, stimulus for what? Well, we have this other structure inside these cells, and it's very much more organized than I'm going to show it here, but it's associated with these Ttubules, and it's the endoplasmic reticulum of the cell. Well, the endoplasmic reticulum does what endoplasmic reticulum does normally, but in contractile cells like skeletal muscle, the endoplasmic, it's called the sarcoplasmic reticulum because sarco refers to muscle, just like myo referred to muscle. Remember the term sarcomere, right? That was the contractile unit. But so they've got a sarcoplasmic reticulum that is this sort of bag. It is a bag, a membrane-bound organelle that stores calcium. Right? And what we're going to find, again, talking about the details of it later, is that this stored calcium needs to be released to stimulate the contraction. Well, again, without getting into too much detail, let's talk about the contractile units. And what that means is we need to sketch, and I'm not going to get into any detail here, this repeating unit of contraction called the sarcomere. And so we're going to have these protein filaments that run lengthwise over and over again. So these red lines are protein filaments. These blue lines I'm gonna make them green actually. Green lines are protein filaments. Right, and these are gonna repeat over and over again. And we're gonna anchor them. And they're gonna repeat. And there's some on this side. And then we repeat the red ones. The red filaments in here. Right, and again, we'll do the structure of this in a separate video. But this is a repeating series of sarcomeres, one after another after another. And they're found in bundles called myofibrils. So we'd have, one here and then another one here and another one over here, right? And again, three-dimensionally within this cell. And those bundles of contractile proteins are called myofibrils. And the repeating unit within a myofibril, like the trains like the cars in a train, right, are the sarcomeres, sarcomere, 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 one after another along the length of this myofibril train, if you will. And they all, will contract and pull on one another to shorten the muscle as a whole. And we'll see that again later. Okay, so we've got our innervation. We've got our Ttubules. The Ttubules exist because these cells are big in diameter as well. And we need to get our stimulus deep into the cell, essentially, where these contractile components are found. So we get to get the stimulus down here so we can release calcium down here. Right? If you only release calcium up here at the surface, we didn't have Ttubules, then these sarcomeres down in the middle of the cell would never receive the calcium stimulus they need to contract and generate tension. So it's essential to have these Ttubules because they help deliver the stimulus deeper into the cell than you'd otherwise find. Okay. One other thing I wanted to talk about, right? I think we've got most of our, and don't forget, we've got our usual, well, I shouldn't say usual. Usual, yes, we have the nuclei and the mitochondria, but remember I said these guys are formed by cell fusion. There's actually another cell type that sort of sits in the periphery. They're called myoblasts. Now, during development, it's the myoblasts that are combining and combining and combining by the hundreds to form these guys. There are some left over post-development that help with repair. They can actually fuse with muscle fibers that have become damaged to repair. And actually, in response to certain types of athletic training, the myoblast can be stimulated to fuse and sort of add new nuclear DNA and new increased ability to generate the billions of copies of the contractile proteins, right? And that's really the whole premise. Well, it's one of the premises of this cell fusion is that, we need to make literally billions of copies of these contractile proteins. Well, proteins are made from DNA. And if you only had one nucleus, you'd be making lots and lots of copies from one source and then have to distribute them throughout the whole cell. Well, instead, we have nuclei, 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 nuclei. Like they're all over the place. And the other thing you have all over the place are the mitochondria. And many of the mitochondria, not all, but many of the mitochondria, they've got two membranes. Many of the mitochondria are sort of found, it's called subsarcolemmal. The sarcolemma is the membrane. Label that. In other words, we don't just call it the cell membrane in a muscle cell. We call it the sarcolemma, and that's the specific name. The more generic name is plasmalemma. That means plasmalemma and cell membrane are the same thing. It's sort of like the fancy term for cell membrane. And so you can say muscle cell membrane, but oftentimes you might hear me say and read it, the muscle cell membrane called the sarcolemma. Right? In other words, this action potential we talked about before is a sarcolemmal action potential. Right? And so we call these mitochondria subsarcolemmal mitochondria. And the idea is this is where we make a lot of the ATP for contraction because these mitochondria are close to the surface where our oxygen supply is coming in, right? Remember, we'd have associated capillaries on the outside of this cell, and that oxygen would be diffusing directly to the cell. Well, the first thing it's going to run into then would be these subsarcolemmal mitochondria, so that we can make ATP very readily as soon as that oxygen sort of comes over the border, if you will, it diffuses into the cell. Okay, I did want to say one other thing about sort of broader organization, and that is, what is the organization of the muscle cells? So the one I drew here, and I'm going to label it, is where the muscle cells run the length of the muscle. They run, well, I should say, they run the length of the muscle, but they run parallel to the length of the muscle, right? So the muscles run in this way, right? It's attached here to here, and the muscle cells are running this way, end to end. And that's called fusiform. And it has a parallel arrangement of muscle fibers. I'll give you the alternative organization, and then we can talk about, just for a minute, about the benefits of one versus the other. The other form of muscle is called penate. And in that form, or in that particular arrangement, the tendon often runs down the middle. Let's have a tendon down the middle. Here it is going to the muscle on one end, or sorry, to the bone on one end, and the bone on the other end. And what happens is, so we've got our muscle sort of suspended in between here, right? Here's the belly of our muscle. But now the fibers are not running lengthwise the way they did up here. Sorry, I just drew it perpendicular just to have more room. But now the fibers on this side are going to run like this. They are not running the full length of the muscle. They are running sort of at an oblique angle to the muscle itself. And so if you think about it, it kind of looks like a feather, doesn't it? Right? Well, the term for feather-like is penate. P-E-N-N-8-A-T-E. Right? So we have fusiform muscles where the muscle fibers are running parallel to the muscle itself, and penate where they're running at an angle to the whole muscle. And here are the benefits of each. In fusiform, the range of motion is very large. However, the total number of cells is reduced, right? Because each cell runs the full length. Each cell is longer, but because each one's longer, you don't, in other words, to form the same size muscle or mass muscle, you need fewer cells to form fusiform, right? Because each one is longer. So what we get out of fusiform muscles is a great range of motion, right? However, if we go to penate, because the muscle fibers aren't pulling in line with the muscle itself, the range of motion is reduced. So you might say, well, what's the plus side then? Well, the plus side is that there are many more cells, which mean, and the strength of a muscle, as we will see in other videos, the strength of a muscle is based upon the cross-sectional area of the cells within it. Because this guy has more cells, it has more what's called physiological cross-sectional area, has more cross-sectional area containing these thick and thin filaments, and so these muscles can generate more force. So more penate muscles, more force, shorter range of motion. Fusiform muscles, less force, but longer ranges of motion, right? So think about muscles in your arms that are like generating these enormous angle changes at your joints, right? We're going to want fusiform muscles to do that. And you may have a combination of those muscles at particular joints, right, where some are there to add more strength, but not necessarily work over the full range of motion of the joint itself. Okay, then we got our two different arrangements of whole muscles. We got our connective tissues mixed in here, and how we have to convey our force from sarcomeres through proteins we don't have drawn here to the extracellular proteins of the connective tissues, and then onward to our tendons, right? And then we talked about the importance of the Ttubules for conveying our stimulus deep into the cell. We're going to link, again in another video, the action potentials in our Ttubules to release of calcium from the, didn't label it, from the sarcoplasmic reticulum, right? Also known as what I'll refer to as the SR, right? That's that guy. And then, of course, that calcium is going to interact with proteins of the found in the myofibrils, the tubes of repeating sarcomeres. And again, the sarcomeres are like a single car in the train, which is this long bundle of fibers called the myofibril.